Welcome to Chapter 6 of Class 9 Science, Tissues. In this fascinating chapter, we'll explore how cells work together in groups called tissues to perform specialized functions in multicellular organisms. We'll discover why plants and animals have different types of tissues, examine the growth tissues in plants, learn about protective and transport tissues, and explore the four main types of animal tissues. Get ready to understand how division of labor at the cellular level makes complex life possible. Let's begin by understanding why plant and animal tissues are fundamentally different. Plants are stationary organisms. They don't move to find food or mates. This sedentary lifestyle means they need large amounts of supportive tissue, often with dead cells that provide structural strength. Their growth is limited to specific regions called meristems. Animals, on the other hand, are mobile. They move to find resources. This active lifestyle requires mostly living tissues, uniform growth throughout the body, and tissues specialized for rapid movement and response. These differences reflect their completely different survival strategies in nature. Now let's explore meristematic tissue, the growth engine of plants. Unlike animals, plants grow only in specific regions where meristematic tissue is located. These tissues contain actively dividing cells characterized by dense cytoplasm, thin cellulose walls, prominent nuclei, and no vacuoles. We have three main types: apical meristem at root and shoot tips for length growth, lateral meristem in the cambium for girth growth, and intercalary meristem near nodes for localized growth. The onion root experiment beautifully demonstrates how cutting root tips stops growth because we remove the meristematic tissue. Let's examine simple permanent tissues in plants, the specialized tissues that result from cell differentiation. When meristematic cells stop dividing, they take up permanent shapes and functions. Parenchyma is the basic packing tissue with thin walls and large intercellular spaces, functioning in storage and photosynthesis. Collenchyma provides flexible support with irregular wall thickenings, allowing bending without breaking. Sclerenchyma offers rigid support with thick lignified walls, making plant parts hard and stiff, like the coconut husk. Observing stem sections under microscope reveals these different cell types and their arrangements. Now let's explore the protective tissues of plants. The epidermis forms the outermost protective layer, a single layer of flat cells with no intercellular spaces, often with a waxy cuticle to prevent water loss. Stomata, surrounded by guard cells, allow gas exchange and transpiration. In older plants, cork replaces the epidermis, consisting of dead cells with suberin in their walls that make them impervious to gases and water. The Rio leaf experiment lets us observe epidermis directly, revealing how this delicate layer protects the plant while allowing essential gas exchange. Let's discover the complex permanent tissues, xylem and phloem, which form the plant's transport system. Xylem conducts water and minerals upward from roots to leaves through tracheids and vessels, mostly dead cells with lignified walls. Phloem transports food throughout the plant through sieve tubes and companion cells, mostly living cells. This vascular system, arranged in bundles, enabled plants to colonize land by solving transport problems. The partnership between xylem and phloem ensures that water, minerals, and food reach every part of the plant efficiently. Now let's turn to animal tissues, beginning with epithelial tissue, the body's protective covering. Epithelial tissue forms barriers that cover organs and line cavities, with cells tightly packed in continuous sheets. We have several types. Squamous epithelium with flat cells for rapid diffusion in blood vessels and lungs. Cuboidal epithelium for secretion and absorption in glands and kidney tubules. Columnar epithelium with tall cells for absorption in the intestine, and ciliated epithelium that moves mucus in the respiratory tract. Each type is perfectly adapted to its specific location and function. Let's explore connective tissues, the most abundant tissues that connect, support, and bind other tissues. Connective tissues have cells spaced apart in an extracellular matrix that determines their properties. Blood has a fluid matrix for transport, bone has a hard matrix for support, cartilage has a flexible matrix for cushioning, and a relar tissue has a gel-like matrix for packing. Specialized types include ligaments connecting bones, tendons connecting muscles to bones, and adipose tissue storing fat. Observing blood under microscope reveals the different blood cells suspended in plasma. Finally, let's examine muscular and nervous tissues, the systems for movement and communication. Muscular tissue consists of cells that contract using special proteins. We have three types: striated muscle for voluntary movement, smooth muscle for involuntary organ contractions, and cardiac muscle for heart beating. Nervous tissue contains neurons that transmit electrical impulses rapidly. The combination of nerve and muscle tissue enables animals to respond quickly to stimuli, the fundamental characteristic of animal life that distinguishes them from plants. As we conclude this chapter, let's review the key concepts and their importance. We've learned about tissue organization in plants and animals, explored how different tissues enable division of labor, understood structural adaptations for specific functions, and appreciated how tissue specialization makes complex life possible. From the growth of plants to the movement of animals, from transport systems to communication networks, tissues represent the next level of biological organization above cells. This knowledge helps us understand how organisms function as integrated wholes and forms the foundation for studying anatomy, physiology, and medicine.